Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimate to you. In the last session, we end up with abnormalities in absorption of lipids. In today's session, we will start with fate of chylomicrons, in which free fatty acids are oxidized to get energy. Then we have beta oxidation of fatty acids, in which two carbon atoms of fatty acids are sequentially cleaved. Lastly, we have a role of carnitine as a transporter across mitochondrial membrane. Good morning everyone. Let us start the topic. Fate of chylomicrons. The absorbed exogenous triglycerides are transported in blood as chylomicrons. They are taken up by adipose tissue and liver. Liver synthesizes endogenous triglycerides. Then they are transported as VLDL or very low density lipoproteins and are deposited in adipose tissue. During starvation states, triglycerides are in adipose tissue are hydrolyzed to form free fatty acids. In the blood, they are transported complexed with albumin. These free fatty acids are taken up by the cells and are then oxidized to get energy. In this diagram, we can see fate of fats in generation of ATP. Liver first releases VLDL with triacylglycerol into the blood, which is taken up by adipose tissue, muscle tissue, and peripheral tissue to generate ATP. On the other hand, hydrolysis to triacylglycerol taken up by chylomicrons, then transferred to adipose tissue, muscle tissue, and peripheral tissue to generate ATP. Coming to beta oxidation of fatty acids. This process is known as beta oxidation because the oxidation and splitting of two carbon units occurs at the beta carbon atom. The oxidation of the hydrocarbon chain occurs in sequential cleavage of two carbon atoms. Coming to preparative stage for beta oxidation. The coenzyme A is a complex molecule containing B complex vitamin pantothenic acid and a molecule of beta mercaptoethanolamine. This SH group from thioester bond in acetyl CoA. To emphasize the function of the SH group, the CoA is sometimes written as CoASH. In this diagram, we can see structure of coenzyme A. It is written as beta mercaptoethanolamine plus 1 beta alanine plus pantoic acid plus ADP. Preparative step 1. Activation of fatty acids. Fatty acids are activated to their coenzyme A derivative. This activation taking place in cytoplasm. ATP gives AMP plus PPI and the energy from the hydrolysis of PPI drives the reaction forward. Thus, two high energy bonds are utilized in this reaction. The enzyme is thiokinase or fatty acyl CoA synthetase. So acetyl group and acyl group are different. Three different enzymes, one each for short chain, medium chain, and long chain fatty acids have been identified. Small chain fatty acids may also be activated by thiophorase enzyme using succinyl CoA. In this diagram, we can see beta oxidation of fatty acids. A fatty acid is converted to fatty acyl CoA by the enzyme fatty acyl CoA synthetase or thiokinase and conversion of ADP to AMP plus PPI. Then this fatty acyl CoA is transferred across the mitochondrial membrane to the mitochondrial side. Then fatty acyl CoA is converted into alpha beta unsaturated fatty acyl CoA with the help of an enzyme acetyl CoA dehydrogenase and conversion of FAD to NADH2. Then this alpha beta unsaturated fatty acyl CoA is converted into beta hydroxy fatty acyl CoA with the help of enzyme enoyl CoA reductase and 1 H2O. Further, this beta hydroxy ketoacyl CoA is converted into beta keto fatty acyl CoA with help of enzyme hydroxy ketoacyl beta hydroxy fatty acyl dehydrogenase and conversion of NAD plus to NADH. Then this beta keto fatty acyl CoA is broken down into fatty acyl CoA plus acetyl CoA with the help of the enzyme thiolase and addition of one CoA SH. This fatty acyl CoA is made to repeat other further cycles, and that belonging to the acetyl CoA goes to the TCA cycle. Acetyl CoA is a combination of acetate or acetic acid, that is 2 carbon units, with coenzyme A. On the other hand, acetyl CoA means acyl group with any fatty acid C4 to C26 in length combined with coenzyme A. Coming to preparative step 2, role of carnitine. Fatty acids are activated in the cytoplasm, but the beta oxidation is in mitochondria. So the transfer of fatty acids through the mitochondrial membrane is essential. The long chain fatty acyl CoA cannot pass through the inner mitochondrial membrane. Therefore, a transporter, carnitine, is involved in transfer of fatty acids. Carnitine is beta-merc hydroxy gamma trimethyl ammonium butyrate. It is written as CH3 thrice Na plus CH2 CHOH CH2 COOH. It is synthesized from liver and methionine in liver and kidney. Preparative step 3, carnitine acyl transferase. The enzyme carnitine acyl transferase 1, that is CAT1, will transfer the fatty acyl group to the hydroxyl group of carnitine to form acyl carnitine. The reaction occurs on the cytosolic side of inner mitochondrial membrane. In this diagram, we can see role of carnitine in transport of acyl groups. Acyl CoA with carnitine forms acyl carnitine. Then this acyl carnitine is transferred across mitochondrial membrane to the mitochondrial side, where it is released into carnitine and acyl CoA. The next is preparative step 4, that is translocase. A protein translocase will carry the acyl carnitine across the membrane to the matrix of mitochondria. On the matrix side of the membrane, another enzyme, carnitine acyl transferase 2, that is CAT2, will transfer the acyl group back to the coenzyme A molecule. Carnitine is returned to the cytosolic side by the translocase. Coming to clinical applications. The medium chain and short chain fatty acids do not require carnitine for transport across inner mitochondrial membrane. So their medium chain and short chain fatty acids are easily oxidized. Carnitine deficiency is reported in preterm infants in which impaired fatty acid oxidation is noticed. More oxidation, glucose is utilized, resulting in episodes of hypoglycemia. Deficiency of translocase. It leads to the defective metabolism of long chain fatty acids. In this condition, muscle cramps are precipitated by fasting, exercise, and high fat diet. Inherited CPT1 deficiency affects only the liver, resulting in reduced fatty acid oxidation and ketogenesis with hypoglycemia. CPT2 deficiency affects primarily skeletal muscle and when severe, the liver. The sulfonylurea drugs, that is clibendilamide and tolbutamide, used in the treatment of type 2 diabetes mellitus, leads to decreased fatty acid oxidation and therefore hyperglycemia by inhibiting CPT1. Coming to beta oxidation steps. The next four reactions are sequentially repeated for complete oxidation of fatty acids. After one round of four metabolic steps, one acetyl CoA unit is uh, split off and acyl CoA with two atoms carbon less is generated. This would undergo the same series of reactions again. The fatty acid is completely oxidized. Coming to step 1, FAD linked dehydrogenase. The free fatty acyl CoA is dehydrogenated to a trans-enoyl CoA with the help of FAD accepting the hydrogen atoms. FADS2, when oxidized in electron transport chain, will produce 1.5 ADP molecules. In this diagram, we can see fatty acyl CoA is converted into alpha beta unsaturated fatty acyl CoA with the help of the enzyme acyl CoA dehydrogenase and the conversion of FAD to NADH2. The next step is hydration. This is catalyzed by enoyl CoA hydratase. This step forms a beta hydroxy fatty acyl CoA. The L isomer alone is formed during the hydration of trans double bond. In the diagram, we can see alpha beta unsaturated fatty acyl CoA is converted into beta hydroxy fatty acyl CoA with the help of enzyme enoyl CoA hydratase and one H2O molecule. Then we have NAD plus dependent dehydrogenase. The beta hydroxy fatty acyl CoA is again oxidized to form beta keto fatty acyl CoA. This dehydrogenase acts only on L isomer. The NADH, when oxidized in electron transport chain, will generate 2.5 ATPs. In this diagram, we can see beta hydroxy fatty acyl CoA is converted into beta keto fatty acyl CoA with the help of enzyme beta hydroxy fatty acyl CoA dehydrogenase and conversion of NAD plus to NADH. Then we have cleavage. The beta keto fatty acyl CoA now undergoes thiolytic cleavage, so that is splitting off a molecule of acetyl CoA and leaving behind a fatty acid with two carbon atoms less. Here we can see beta keto fatty acyl CoA which is converted into fatty acyl CoA and acetyl CoA with the help of the enzyme thiolase and CoASH. Here ends the session. Kindly like, share and subscribe the channel. Thank you.